then that will give you time. There's a PhysioX lab assigned. We will work on the wet lab. We have our rabbit muscle lab tomorrow, which is part of that lab. Not a dissection. The fibers are already taken out. It's looking at the contraction in the microscope, adding different chemicals and seeing if we can see that sarcomere shorten. And then the 2 labs are due next week. And so because of that, because you guys are still working on your take-home exam, that dirty word, 3 words, so you can get started on the PhysioX, you can work on the take-home. There is a lab quiz tomorrow. The lab quiz is on Tuesday's lecture. So the structure of a muscle cell, the terminal cisternae, sarcoplasmic reticulum, sarcolemma, epimysium, all of that. We have a couple of models over there that it helps you to see those structures in 3 dimension, and that will be tomorrow's quiz. So today's lecture is moving into the physiology. So except for what we review on the sarcomere, the physiology part of it won't be on the quiz. I did turn in a new order for the second part of the lecture notes. Remember I said there was another part and the pictures were overlapping and everything. So I turned it in and it should be here on Monday. So I'll have it for you Tuesday and we'll start the identification of muscle structure and functions. All right, so up on the board is an image of a microscopic view of a sarcomere. And let me just bring up one of these models here. I just finished grading your last exams, and I'll total them up and post them today. So on the outside of this, we have the plasma membrane of a muscle cell. Do you remember what that plasma membrane is also called? No, that's the connective tissue. So another name, we had 2 roots for muscle, myo and sarco. Excellent. And so we had axolemma and neurolemma, so our root for membrane is lemma. So sarcolemma is the other name for the plasma membrane surrounding a muscle cell. This is the loose irregular connective tissue around an individual muscle cell, so that would be the endomysium, all right, around each individual muscle cell. And then what's our other word for a muscle cell? Myofiber, okay? Myofibril, remember, are these bundles of myosin and actin that are actually within the muscle cell. So here's the sarcolemma, and then you can see these darker orange structures here are the invaginations of the sarcolemma deep into the muscle cell, carrying the action potential. We're going to talk about that process today. And these are known as the transverse tubules. So they carry, yes, the T-tubules. They carry a channel, a tube of plasma membrane deep into the muscle cell so that electrical current can follow it in there as well. Then, on either side of each transverse tubule, we have a modified smooth endoplasmic reticulum which stores calcium ions. And so instead of smooth endoplasmic reticulum, we use that sarco term again, and it's called the sarcoplasmic reticulum. All right? Just adjacent to the transverse tubule, the sarcoplasmic reticulum is dilated, having voltage-gated channels that are lower in their threshold than the rest of the sarcoplasmic reticulum, and those were the terminal cisternae. Okay, cisternae meaning like a dilated sac. Alrighty, so those are essentially the components of a skeletal muscle cell. Now you'll notice that there is banding on this cell. So the myofibril, each of these is a bundle of actin and myosin, which is the myofibril. And the thick ones are myosin, and the thin ones are actin, and they have a distinct overlapping pattern to them that when we look at the outside of the muscle cell, we see as dark and light stripes. So that's what I've shown up on the board and what I wanted to review with you before we move back into the actual muscle contraction. But that was the entire lecture that you missed on Tuesday in 2 minutes.
Oh, wow. Sounds so easy.
So if we look at this online, you can see that we have the dark region, and then we have the light region, all right? The dark region is dark because it's determined by the length of the thick filament, which is myosin. Does that get shorter when a muscle cell contracts?
No.
No, okay? Now notice that the white band requires parts of two sarcomeres. Our sarcomere is the shortest unit of contraction and is determined from Z disc to Z disc, while our I band is on either side of the Z disc. So I want to re-diagram that, put up on the board what structures change and what structures don't change when a muscle cell contracts. And this is just a review of what we covered on Tuesday, so you can listen, you can draw it again, that's kind of up to you. So here's the myosin. I'm not going to add in the myosin heads. And then we'll draw another set of myosin over here. And then we have the actin attached to the Z disc. So from Z disc to Z disc, and it's called a Z disc because the proteins that attach each actin kind of go in a zigzag formation here. So this is the Z disc or Z line. And that determines the sarcomere. Alright, so when a muscle changes, when a myofiber goes from a relax to contraction, is the sarcomere going to shorten or not?
Yes.
Will the distance between the Z disc change? Yes. So the sarcomere will shorten or narrow. Remember, that's our smallest unit of contraction. So all of the sarcomeres will come closer together. Within a sarcomere, so within a sarcomere, the dark band is the width of the myosin, and this was called the A band, if you recall. Right? Does the A band narrow?
Yes.
This from here to here, will that become narrow when the muscle cell contracts? No, because that would require the myosin filament getting shorter. The myosin filament does not get any shorter. All right? What's happening is actin overlaps it more. It might get darker, but it doesn't get shorter. All right? So the A band is unchanged. Now within the A band, we have a lighter area, what you see here, which is lighter because actin is not overlapping it. So here actin is barely overlapping the myosin. So we have a very wide H band or H zone. Either one is fine. So as the actin and actin filaments are pulled over the myosin, that is going to become more narrow. Here it's almost disappeared. Here the actin filaments are actually overlapping each other, and the H band's completely disappeared. So with contraction, the H band will narrow. Yes?
when there's a contraction of, they don't like ratchet down like this, do they? Each section just kind of goes in once?
No, well, I'm not sure what you mean by a ratcheting. I'm going to kind of illustrate it for you. There is kind of a hand over hand pulling of the actin. The myosin, one myosin head will bind to the actin,
Yeah.
swing forward. Another one will bind, swing forward. This one will let go, bind
Same
again.
exact section?
Yeah.
So they alternate.
So they
kind of,
keep-
don't
well, there's
all go
thousands of
together
them.
and then back out.
Right. There's thousands of them, so they're all of them doing that.
So they just at different times.
Right.
Because I was trying to figure out if they all went in together and then
No.
and then
They
some
let
had
go.
went out and went back in.
Because there's always a tension, so there's always some of them attached while others are letting go.
Okay, so they
To
don't do it
pull
all
it.
at the same.
Yeah.
Okay.
All right, so the H-band is going to narrow. And then we remember we had proteins attaching the myosin bands together, the myosin binding components here that form the M line. And obviously that's not going to change. So notice that to form the I band, we take this portion of one sarcomere and this portion of the adjacent sarcomere, and that forms the I band. So an I band requires two adjacent sarcomeres. When the muscle cell shortens, what happens to our I band?
Shorten.
It's also going to shorten or narrow because more of the actin is overlapping the myosin, so there's going to be less actin by itself. Okay, that makes sense, then you're all the way there, okay? You understand what's going on here. Now, here this looks almost as wide as that, even though we have a more narrow H zone. When I show this on quizzes or on lecture exams, I do the same thing. Because if you look at this next picture, if I say, looking at this sarcomere, is the muscle cell contracted or relaxed? Well, here it's shorter, so obviously it's contracted. So I use the computer and I will take this and stretch it out so that the sarcomere is actually the same length and force you to look at the H zone and the I band to see if they have changed, okay? Because otherwise it's just too obvious and the sarcomere shortened, so obviously the muscle cells that have contracted. So I do that artificially. This is the actual view where the sarcomere is shorter. The sarcomere is more narrow. The H band is narrow and the I band is narrow. Okay. We'll come back to this image on the bottom tomorrow. It's just showing the relative, this is not a depolarization, repolarization graph. This is a force, this is increased tension and decreased tension created by the myosin interacting with the actin. So if we start out with the actin completely overlapping the myosin, there's not much more tension that can be produced. If we end up with the actin barely overlapping the myosin, it's going to be really difficult for those myosin heads to attach to the actin, so it's very difficult to create a force. So there's an optimum amount of overlap between actin and myosin to generate the greatest amount of force, and that's really all that's being depicted by this graph. But we'll look at it some more tomorrow. Okay, let's switch to the physiology then. So this is on page 14, and the actually it's page 18. The steps that I'm going to go through are written out for you in sequence on page 22. All right? But I would suggest that you understand the process and not try to memorize these steps. Again, if you understand logically what needs to happen, then the next step will come to you as a logical process rather than, if you're trying to memorize how to count in a foreign language. You're just trying to what's that word? And it doesn't make any sense why it would be the next word. So, but it should make sense in a mechanism what the next logical step would be. So if you understand that, like a little movie going on, that's a lot easier to recall than memorizing a series of steps. So page 22 is a series of steps of contraction, and then on page 24, the steps of relaxation. Okay? May I erase the board? Okay.
So for tomorrow's quiz, it's just going to be labeling the zones.
And the IBAN, yeah, labeling this, labeling the parts on the muscle cell, yeah. Huh? I'll try not to break a leg. Okay.
What does the bottom right say, the Z?
Z-disc.
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 Sorry.
No, it's okay.
It's a list. It's a new word.
At least quick.
From the back of my head, the whole time I'm lecturing you guys, I'm thinking about my daughter who has her 300 question test on identifying all 600 muscles of the body.
Wow.
So they have a Thanksgiving feast today, and she said she had to bring mashed potatoes. And I said, for how many people? And she said 31. And I said, honey, there's 31 people in your class. If everyone brings food for 31 people, you're going to have way too much food. So I'm sure some other people, you know, you're having 2 or 3 people bring this, 2 or 3 people bring that. So I got the mashed potatoes ready in a big bowl, and as I headed out the door to the garage, I said, don't forget the mashed potatoes.
She forgot.
And she forgot.
So we're halfway to school, and she goes, Mom, I forgot the mashed potatoes. So I knew she had this test. I said, honey, don't worry about it. There'll be plenty of food. Even if nobody else brings mashed potatoes, they'll have plenty of food to eat. Don't go, she has the car. I said, don't take the car and go buy more mashed potatoes. Just forget it. Apologize and move on. All right, so this is back to the diagram, the model that I have on the table, okay? And this is where we're going to look at the physiology of a muscle contraction. We're going to do a little bit of review, at least review in the sense of we're going to be talking about ion channels and events that we've already covered. This is why we did this is why we did nerve physiology first. We're going to follow that action potential to the muscle cell. So here we have a lower motor neuron sending axon terminals. This is only showing one muscle cell, all right? Well, actually, it's showing with all the myofibrils here. And one muscle cell is actually going to one neuron is going to innervate multiple cells. Here it shows just 1 to 1. I don't know of any case in the body where we have such a fine motor control that we have only one neuron for one muscle cell. A low ratio would be maybe 1 neuron to 5 or 10 muscle cells. So in your fingertips and your lips for fine motor movement. In your back, it would be maybe 1 neuron, 1 nerve cell to 100 or 500 muscle cells. And that ratio is called a motor unit. All right, so on the top of page 18, you see the term motor unit. And the definition is a nerve cell and all the muscle cells that it innervates. All right. What we're going to look at more closely right here is the relationship between an axon terminal, its synaptic bulb, and the underlying muscle membrane. So you have this diagram on the bottom of page 18. So here is our lower motor neuron with its axon, synaptic bulb. So this is a lower motor neuron. And it's going to form a relationship with the muscle cell. So here's the sarcolemma. And at the site where it interacts with the synaptic bulb, it is thrown into multiple folds which function to increase surface area. So this membrane of our lower motor neuron is known as the presynaptic membrane. Remember that term? And if this was a neuron, another neuron, we would call it the postsynaptic neuron. The whole thing would be a synapse. Because this is a skeletal muscle cell, instead of calling it a postsynaptic membrane, because we talk about this as a junction rather than a synapse, this is described as the motor end plate. So don't confuse that with a motor unit, all right? The motor end plate is the equivalent of the postsynaptic membrane. And this is a neuromuscular junction. The space, we still come back confusingly and call this space the synaptic cleft, not the junctional space. All right, but it's the distance that our neurotransmitters would have to diffuse. So as we send, I decide that I want to pick up the controls here for the projector. And I reach out my hand, I'm contracting my triceps brachii muscle to reach out, straighten my elbow to pick that up. So in so doing, I'm activating upper motor neurons, which are synapsing in my spinal cord with the lower motor neurons. That's causing a sequence of sodium, potassium, voltage-gated channels to open and sending that signal down my axon terminal. Once that series of action potentials reaches the synaptic bulb, what kind of channels are going to open? Calcium, voltage-gated calcium channels. So this is a voltage-gated calcium channel. Calcium ions enter the synaptic gluton and they will bind, wrong color. They will bind to a calcium binding protein on the surface. Anybody remember what that's called?
Synaptotagmin.
Exactly, synaptotagmin. Let me do that in green. So once the calcium binds to the synaptotagmin, that changes the configuration of the membrane and allows it to fuse with the presynaptic membrane and release the neurotransmitters. So what are the neurotransmitters? Acetylcholine. So acetylcholine is released. What kind of channels is it going to bind to in our motor end plate? Ligand gated or voltage gated? Ligand gated. And you're right, they are nicotinic acetylcholine receptors. So ligand gated acetylcholine receptors, nicotinic, meaning they are ionotropic. Excitatory or inhibitory? Excitatory. See all that stuff that you know already now? Took us a long time to get here, but once we're here. So now this is going to open sodium channels. And sodium ions will enter the cell. Muscle cells also have threshold potential. So this is a graded potential, right? Voltage gated, ligand gated channels, when they open, create graded potentials. Because they can open a lot, many times, or a few times, or multiple times. And so we're creating a graded potential. And as these ions enter the cell, they are going to flow along the cell membrane. In all directions. And if they reach threshold, they will open voltage-gated channels in the sarcolemma outside the motor end plate. So the motor end plate has ligand-gated channels. The rest of the sarcolemma is going to have voltage-gated channels. All right. So these are voltage-gated channels for sodium and potassium, just like we saw in the nerve cell. A muscle cell is excitatory, okay, and can conduct a current. So these are voltage-gated sodium and voltage-gated potassium channels. If threshold is reached, all right, so if we reach the threshold potential, those channels will open and will start an action potential flowing across the cell membrane of the muscle cell. So almost exactly the same as we saw from a presynaptic neuron to a postsynaptic neuron. Okay? It's just that our voltage-gated channels are much closer to the formation of the graded potential. We don't have a whole cell body that it has to be able to diffuse through to get to the axon. All right? Our voltage-gated channels are just right next door to this motor end plate. Now we start to see a few differences. So as this action potential is traveling down the muscle membrane, it's going to find this depression in the muscle membrane created by the indentation and formation of the transverse tubule. Now, it's a little, just to remind you, each of these holes here is the transverse tubule. So as that current spreads across, it's going to flow around the transverse tubule as well as down it. In other words, that current doesn't have to go down here and then come back up again, because this is just a small hole. We'll go around it and go down this side as well. Does that make sense? So it's going to go down the membrane on both sides because this is so small. It's just like if you have a drain in the shower, the water's not going to go down just one edge of that hole in the shower. It's going to go around the entire drainage and flow into that hole from all sides. So the same thing with our current. So now our action potential is traveling down the transverse tubule. And why can it travel down the transverse tubule? Not only does it have voltage-gated channels, but the content, the fluid of our transverse tubule is still extracellular fluid. So we have the sodium, we have those ions available to continue the perpetuation of that current. All right, so far, nothing different from what we talked about with neurons, as far as how this is all happening. Any questions on this before we now add what's going on inside the muscle cell? If you want to take a picture, go ahead, because I'm going to erase these words and add in the sarcoplasmic reticulum. is illustrated, slightly different picture, but illustrated on the left-hand side, the lower portion of page 20. So we have the transverse tubule associated with the modified smooth endoplasmic reticulum storing calcium ions. So that's what I'm drawing here in black. So this is the sarcoplasmic reticulum. And the dilated region on each side is the terminal cisternae. It has a lower threshold. It doesn't require quite as strong a current for its voltage-gated channels to open. And so as the action potential travels down the transverse tubule, we also will open voltage-gated channels in the terminal cisternae. And these are voltage-gated channels for calcium. So the current of the transverse tubule is sufficient to open the voltage-gated calcium channels along the terminal cisternae. And that creates a current strong enough to open voltage-gated channels throughout the rest of the sarcoplasmic reticulum. And calcium ions flow out of the sarcoplasmic reticulum and enter into the cytoplasm of the cell. Calcium ions are stored in the sarcoplasmic reticulum, and then they leave when the voltage-gated channels are open. And the amount of free calcium in the cytoplasm of the cell increases. So let me down the lights, and this is shown in the upper portion of this diagram. So up here is what we just got through talking about. The action potential reaching the synaptic bulb results in the release of acetylcholine, which binds to ligand-gated receptors, creating a graded potential that reaches the threshold potential of voltage-gated sodium-potassium channels, sending an electrical current that travels down the transverse tubule. And this opens voltage-gated channels along the sarcoplasmic reticulum, and that releases calcium into the cell. Okay? Now we're going to look at what that calcium does. In the synaptic bulb, the calcium bound to synaptotagmin, which allow the vesicles to fuse with the membrane. Inside the cell, the calcium is going to bind to that molecule I told you about on Tuesday called troponin. Remember when we described the actin filament? It had troponin and tropomyosin. So I'm going to go through that process with a diagram here on the screen, and then I'll diagram it on the board for you. So what we're looking at here is, remember actin as a filament was two strands of actin twisted together? So we have this helix. So this is just showing one bead from each strand. So the large purple, blue circles are an actin from each of those two strands. Tropomyosin was a long protein filament that ran along the groove. And here we're seeing it in cross-section, this small orange molecule. And it was held in place by troponin. So these yellow circles here are representing troponin. All right? When calcium ions bind to troponin, it separates from the actin filament. That allows the tropomyosin, which is covering the binding site, to move into the groove between the two strands, and that exposes that binding site and allows that golf club shaped head of the myosin to attach to it. That's the whole role and relationship between calcium and troponin. As soon as we dump calcium into the cytoplasm of the muscle cell, this event is going to occur. At the same time, if we want our muscle cell to relax, what do we have to do? Get rid of the calcium. Okay, so the calcium is required for contraction and its removal is required for relaxation. So you have this diagram at the top of page 20. So with your permission, I'll erase the board. Anybody else? I added, you guys need any of that new material on there? If not, I'll go ahead and erase it. Okay. 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 What I've left unsaid here is that obviously a muscle cell has to have a negative resting potential for all this to work. So the same types of things that allowed a negative resting membrane potential in the nerve, the leak channels and the sodium potassium channels and the proteins with negative charges are going to apply here as well. All right, so here is our double twisted strands of actin. So this would be our filamentous actin. And within each groove here, we have the tropomyosin. And holding that tropomyosin in place, we have the troponin. Okay? Now if I bring in the myosin filament, this would be in a muscle cell that is relaxed. Okay. The myosin is going to have its myosin head. Remember, this is an ATPase enzyme that breaks down ATP. So it is already broken down ATP, and the ATP is in the ABP form. Now I'm just going to draw one myosin filament rather than the protein, rather than the multiple ones with thousands of heads, just to make it a little easier to see. So then the next step is calcium binds to troponin. So calcium ions and form troponin calcium complex, which dissociates from actin. Without troponin, tropomyosin moves into the groove. And as a result of that, myosin binds to actin. Okay, so I'm going to redraw that picture to illustrate these events right here. So troponin is off to the side, bound to calcium ions. So these would be the troponin calcium complexes. Everybody with me? Too busy drawing. The tropomyosin is moved deep into its groove. We can barely see it. And the myosin head is attached to its binding site. That won't happen if calcium isn't released from the sarcoplasmic reticulum.
And the blue dashes on.
Is the tropomyosin that's deep in its groove. So that event is what we saw on the previous screen as the tropomyosin moving from here to here. Okay.
That's the contraction of the muscle.
Not yet, because we haven't moved it yet. Okay. A nice segue though, all of the events that we've talked about so far, from the current moving along the neuron to opening of ligand-gated channels to the current moving into the cell, the calcium being released, all of those first few steps occur before any tension is created. So that's known as the lag or latent period. All those events are having to occur before tension is created. So if you look on page 26, that lag phase, that's what we've gotten to at the end of the lag phase, on page 26. That lag phase or latent period. Okay?
No, you're still talking about, is this, the calcium is what wears down the muscles?
Typically it's ACT, but we'll talk about that. There's 3 regions where ACT can occur. All right, so far we haven't generated any tension. All right, it's going to, we've kind of paused it. It won't not naturally pause here. I paused it just to kind of, okay, this. As soon as myosin binds to the actin, the tension that's caught is released. And that head swings forward. All right, that's the creation. That's the first time we create any tension. So the next event then is the myosin head swings forward, sliding the actin. Towards the M line. So I'm going to move the actin over here slightly. Notice how the position of the myosin head changes. It is now in this position, and so as in so doing, it's pulled the actin, slid the actin towards the center of the A band. So on this diagram, that's what we're seeing right here, a 1 to 2. Okay. So here the head is attached to the actin, but it couldn't do that until the troponin was removed. As soon as it attaches to the actin, and it's doing so with ADP attached to it, as soon as it attaches, it swings forward, and that's the first creation of tension and shortening of the sarcomere. Okay? So let me pause there for just a moment to illustrate what's going on. So these two yardsticks will be the actin filaments. Okay? And my upper extremities will be myosin. And the actin overlaps that to some degree. Okay? In reality, when the muscle cell is relaxed, it's not attached to it. But the myosin head is going to be in this position. When calcium removes the troponin, it binds to it, swings forward. Okay? So the ends of each of these determines the length of the sarcomere, and it keeps swinging it forward and short, bringing more and more of the actin